The retina forms the innermost neural layer of the eyeball, lining the posterior part of the eye where incoming light is ultimately focused after passing through the cornea and lens. Its primary role is to capture light and convert it into neural signals that can be interpreted by the brain. The outermost component of the retina is the retinal pigment epithelium, a layer rich in melanin pigment. This layer absorbs excess scattered light, preventing internal reflection within the eye and thereby helping maintain visual clarity. It also supports photoreceptors metabolically and removes debris from their outer segments, maintaining the functional integrity of the visual cells. Just internal to this lies the photoreceptor layer, which contains the specialized sensory neurons known as rods and cones. These cells capture incoming photons and begin the process of phototransduction, converting light energy into electrical signals. The signals generated in the photoreceptors are then transmitted to the bipolar cells, which serve as intermediary neurons within the retina. Bipolar cells begin the initial processing of visual information, including contrast and light intensity. From bipolar cells, the signals are passed to retinal ganglion cells, whose axons converge at the optic disc to form the optic nerve. Unlike other retinal neurons, ganglion cells generate true action potentials, allowing visual signals to travel efficiently toward the brain. Through this layered neuronal organization, the retina functions not merely as a light detector but as a complex neural processor, performing the first stage of visual interpretation before transmitting the information to the visual centers of the brain. The great vessels are the major arteries and veins that directly connect to the heart, forming the main inflow and outflow tracts. The arteries include the aorta and pulmonary trunk, while the veins include the superior vena cava, inferior vena cava, and 4 pulmonary veins. The aorta is the main systemic outflow vessel arising from the left ventricle. Its first part, the ascending aorta, gives origin to the right and left coronary arteries that supply the heart. It then continues as the arch of aorta at the level of T4, which gives 3 important branches from right to left: the brachiocephalic trunk, left common carotid artery, and left subclavian artery. Beyond this, it continues as the descending thoracic aorta, which passes through the diaphragm at T12 and becomes the abdominal aorta. The pulmonary trunk arises from the right ventricle, runs anterior to the ascending aorta, and divides at the level of T6 into right and left pulmonary arteries carrying deoxygenated blood to the lungs. The ligamentum arteriosum is a fibrous remnant of the fetal ductus arteriosus that connects the left pulmonary artery to the arch of the aorta. The superior vena cava is formed by the union of the right and left brachiocephalic veins and returns blood from the upper body to the right atrium. The inferior vena cava is formed by the common iliac veins, passes through the diaphragm at T8 and carries blood from the lower body to the right atrium. The 4 pulmonary veins, 2 from each lung, carry oxygenated blood to the left atrium. Embryologically, these vessels develop from the truncus arteriosus and the aortic arches, separated by the spiral aorticopulmonary septum during the 5th to 8th week. The 3rd arch forms the common carotid arteries, the 4th arch contributes to the arch of the aorta and right subclavian artery, and the 6th arch forms the pulmonary arteries and the ductus arteriosus. The thalamus is a paired, ovoid mass of grey matter located deep in the diencephalon, forming the lateral walls of the 3rd ventricle. It lies medial to the internal capsule and superior to the hypothalamus, with the 2 thalami sometimes connected by the interthalamic adhesion. Structurally, each thalamus is divided by a Y-shaped internal medullary lamina into anterior, medial, and lateral parts. Postroventrally, the thalamus shows 2 distinct enlargements: the lateral geniculate body, which serves as the visual relay, and the medial geniculate body, which serves as the auditory relay. Dorsally, the thalamus is covered by the stratum zonale, while laterally it is bounded by the external medullary lamina, beyond which lies the thalamic reticular nucleus. Functionally, the thalamus is organized into nuclear groups. The ventral anterior and ventral lateral nuclei are linked to motor control, relaying information from the basal ganglia and cerebellum to the motor cortex. The ventral posterior lateral (VPL) Nucleus relays somatic sensation from the body, while the ventral posterior medial (VPM) nucleus relays sensation and taste from the face. The anterior nucleus connects with the limbic system and participates in emotion and memory. The medial dorsal nucleus integrates emotional, cognitive, and behavioral information with the prefrontal cortex. The reticular nucleus regulates thalamocortical activity and attention. The fibrous outer coat of the eye is formed by the sclera posteriorly and the cornea anteriorly. At the front of the eye, the opaque sclera continues as the cornea, a transparent dome at the corneoscleral junction or limbus, allowing light to enter the eye. The cornea is avascular, meaning it has no blood vessels, which helps maintain its transparency for light transmission. However, it is richly supplied with sensory nerves, making it highly sensitive and therefore acting as a protective warning system for the eye. Structurally, the cornea has 5 layers arranged from anterior to posterior. The corneal epithelium forms the outer barrier, protecting the eye from dust and microbes. Beneath it lies Bowman's layer, providing structural support to maintain the corneal curvature. The stroma, the thickest layer, contains organized collagen fibers that preserve transparency and strength. Behind it lies Descemet's membrane, supporting the inner corneal surface. Finally, the endothelium regulates fluid balance, preventing corneal swelling and maintaining clarity. Together, this arrangement allows the cornea to act both as a protective shield for the eye and as the primary refractive surface, bending incoming light before it reaches the lens to help produce clear and focused vision.
Ventricular systole marks the precise conversion of intraventricular pressure into effective arterial blood flow. As ventricular contraction continues, the pressure inside the ventricles rises rapidly and soon exceeds the pressure in the aorta and pulmonary trunk. This pressure difference causes the semilunar valves, the aortic and pulmonary valves, to open, marking the beginning of the ventricular ejection phase. Blood is then ejected forcefully from the ventricles into the great arteries. The left ventricle pumps oxygenated blood into the aorta for systemic circulation, while the right ventricle pumps deoxygenated blood into the pulmonary trunk toward the lungs. This phase represents the main pumping action of the heart and is essential for maintaining effective circulation. During ventricular ejection, ventricular volume decreases significantly as blood leaves the chambers. However, a small amount of blood remains in the ventricles after contraction, known as the end systolic volume. The amount of blood ejected during each heartbeat is called the stroke volume, and it plays a crucial role in determining cardiac output. The efficiency of ventricular pumping is measured clinically by the ejection fraction, which reflects the percentage of blood ejected from the ventricles. As ventricular contraction ends, the ventricular muscle begins to relax, causing intraventricular pressure to fall rapidly. When ventricular pressure becomes lower than the pressure in the aorta and pulmonary trunk, blood briefly flows backward toward the ventricles, filling the cusps of the semilunar valves and causing them to close. This produces the second heart sound, S2, and marks the end of ventricular systole. The ventricles then enter isovolumetric relaxation, during which all valves remain closed and ventricular pressure decreases without a change in volume. When ventricular pressure falls below atrial pressure, the atrioventricular valves open, allowing passive filling of the ventricles. This initiates ventricular diastole and prepares the heart for the next cardiac cycle, ensuring continuous and efficient blood circulation. Every heartbeat is a precisely timed mechanical and electrical event that sustains life. The cardiac cycle is defined as the sequence of alternating contraction and relaxation of the atria and ventricles that enables the heart to pump blood efficiently throughout the body. It begins at the start of one heartbeat and ends at the beginning of the next, ensuring continuous circulation through the pulmonary and systemic systems. The cardiac cycle consists of 2 major phases: diastole, the relaxation and filling phase, and systole, the contraction and ejection phase. These phases work together to maintain effective cardiac function and continuous blood flow. The cycle begins with ventricular diastole, during which the ventricles are relaxed and filling with blood. Blood returns to the right atrium through the superior and inferior vena cava and to the left atrium through the pulmonary veins. As blood accumulates in the atria, atrial pressure increases. When atrial pressure exceeds ventricular pressure, the atrioventricular valves, the mitral and tricuspid valves, open, allowing passive flow of blood into the ventricles. This passive filling accounts for the majority of ventricular filling. The sinoatrial node then generates an electrical impulse which spreads across the atrial myocardium, causing atrial depolarization and atrial systole. Atrial contraction pushes the remaining blood into the ventricles, ensuring complete ventricular filling. The volume of blood present in the ventricles at the end of this phase is called the end-diastolic volume, which represents the maximum ventricular filling before contraction. Following this, ventricular systole begins with ventricular depolarization, represented by the QRS complex on the ECG. As the ventricles contract, intraventricular pressure rises rapidly, forcing the atrioventricular valves to close and producing the first heart sound, S1. At this moment, the semilunar valves remain closed and the ventricles contract without a change in volume. This phase is called isovolumetric contraction and allows pressure to build within the ventricles before blood is ejected.
Conduction system of the heart is a specialized network of modified cardiac muscle fibers responsible for initiation and propagation of electrical impulses, ensuring coordinated atrial and ventricular contraction. The impulse normally originates from the sinoatrial node, located in the right atrium near the opening of the superior vena cava. It is the primary pacemaker of the heart with an intrinsic rate of 60 to 100 beats per minute. From the sun node, impulses spread through the atrial myocardium via internodal pathways and through Buchmann's bundle to the left atrium, producing synchronous atrial contraction. The impulse then reaches the atrioventricular node, situated in the lower interarterial septum within the triangle of Koch. The AV node produces a physiological delay, allowing adequate ventricular filling before contraction. From the AV node, the impulse enters the atrioventricular bundle, also known as the bundle of His, which forms the only electrical connection between the atria and ventricles. The right ventricle can be identified by its characteristic internal features as blood flows from the right atrium into it and then to the lungs. Its inner surface shows irregular muscular ridges called trabeculae carni, giving it a sponge-like appearance. It contains 3 papillary muscles, anterior, posterior, and septal, connected to the tricuspid valve by chordae tendineae. The smooth outflow tract of the right ventricle, known as the conus arteriosus or infundibulum, leads into the pulmonary trunk. A prominent muscular band called the septomarginal trabecula, or moderator band, carries part of the conduction system across the cavity. On the left side, the left atrium forms the base of the heart and receives oxygenated blood from 4 pulmonary veins with a small tubular auricle. The left ventricle has much thicker muscular walls than the right because it must generate high pressure for systemic circulation. It contains 2 large papillary muscles supporting the mitral valve and shows finer, more numerous trabeculae carni. Clinically, defects at the site of the fossa ovalis can lead to atrial septal defects with left-to-right shunting. The left ventricle may hypertrophy in chronic hypertension or aortic stenosis. The right ventricle may hypertrophy in pulmonary hypertension, and lesions near the crista terminalis can affect the sinoatrial node and disturb normal heart rhythm. The heart consists of 4 chambers working in a coordinated flow to maintain circulation. The right atrium is the chamber that receives deoxygenated blood from the superior and inferior vena cava. Structurally, it has 2 distinct regions: a smooth posterior part called the sinus venerum, which develops from the sinus venosus, and a rough anterior part containing pectinate muscles derived from the primitive atrium. These 2 regions are separated internally by a C-shaped muscular ridge known as the crista terminalis. At the upper end of this ridge, near the opening of the superior vena cava, lies the sinoatrial node, the natural pacemaker responsible for initiating the heartbeat. The interarterial septum, which separates the 2 atria, shows a thin oval depression called the fossa ovalis. This structure represents the remnant of the fetal foramen ovale, which allowed blood to bypass the lungs before birth, and it is surrounded by a raised margin known as the limbus fossa ovalis. The wall of the heart is organized into three histological layers: epicardium, myocardium, and endocardium, and the entire heart is enclosed by the pericardium. The epicardium forms the outermost layer of the heart wall and is also known as the visceral layer of the serous pericardium. It is composed of a thin lining of simple squamous mesothelial cells, supported by connective tissue and variable amounts of fat. This layer is clinically important because it contains the coronary arteries and veins, lymphatic vessels, and autonomic nerves supplying the heart. Beneath the epicardium lies the myocardium, the thick muscular middle layer responsible for the pumping action of the heart. It is composed of cardiac muscle cells that are striated, branched, and interconnected by intercalated discs, which provide mechanical strength and allow rapid electrical conduction for coordinated contraction. The thickness of the myocardium varies in different chambers, being greatest in the left ventricle, followed by the right ventricle and then the atria, reflecting the workload of each chamber. Damage to this layer due to reduced blood supply leads to myocardial infarction. The endocardium forms the innermost smooth lining of the heart chambers and cardiac valves. It consists of simple squamous endothelial cells supported by a thin connective tissue layer, with a deeper subendocardial layer containing Purkinje fibers, which are part of the cardiac conduction system and help regulate the heart's rhythm. The cranial part of the neural tube expands to form three primary brain vesicles: prosencephalon, mesencephalon, rhombencephalon. These further subdivide into 5 secondary vesicles: telencephalon, cerebral hemispheres; diencephalon, thalamus, hypothalamus, epithalamus; mesencephalon, midbrain; metencephalon, pons and cerebellum; myelencephalon, medulla oblongata. The central lumen of these vesicles gives rise to the ventricular system, including the lateral ventricles, third ventricle, cerebral aqueduct, and fourth ventricle, which remain essential for CSF circulation. In the spinal region, the neural tube organizes into ulnar plates, sensory, and basal plates, motor, separated by the sulcus limitans. Neural stem cells from the ventricular zone proliferate and differentiate into neurons and glial cells. Subsequent processes include axon guidance, synaptogenesis, and myelination, the latter continuing well into postnatal life. Thus, within a few weeks, coordinated molecular and morphogenetic events establish the basic architecture of the CNS and PNS. Disruptions during this window result in significant congenital neurological anomalies. Neural impulses generated by rods and cones are transmitted to the brain through the optic nerve, cranial nerve 2. The optic nerve is formed by the axons of retinal ganglion cells, which converge at the optic disc. Hence, the optic disc has no photoreceptors and represents the physiological blind spot. The optic nerve exits the orbit through the optic canal and enters the cranial cavity. At the optic chiasm, located superior to the pituitary gland, there is partial decussation of fibers. Fibers from the nasal retina cross to the opposite side, while fibers from the temporal retina remain uncrossed. This arrangement is essential for binocular vision and explains field defects seen in chiasmal lesions, example bitemporal hemianopia. Beyond the chiasm, fibers continue as the optic tracts, which carry visual information from the contralateral visual field of both eyes. Most fibers of the optic tract terminate in the lateral geniculate nucleus, LGN, of the thalamus, the primary relay station for vision. From the LGN, fibers pass as optic radiations, geniculocalcarine tract, to the primary visual cortex, Brodmann area 17, located along the calcarine sulcus of the occipital lobe. A small proportion of optic tract fibers bypass the LGN and terminate in the superior colliculus, visual reflexes and eye movements, and pretectal nucleus, pupillary light reflex, which is clinically important in assessing afferent pupillary pathway lesions. The cerebellar cortex is grey matter surrounding central white matter and is organized into 3 distinct layers, uniform throughout the cerebellum. The molecular layer is the most superficial, lying just beneath the pia mater. It contains dendritic arbors of Purkinje cells, parallel fibers from granule cells, and inhibitory stellate and basket cells. This layer is primarily a site of synaptic integration. Beneath it lies the Purkinje cell layer, a single row of large Purkinje neurons. These cells form the only efferent output of the cerebellar cortex and project inhibitory signals to the deep cerebellar nuclei and vestibular nuclei. The deepest layer is the granular layer, densely packed with granule cells and Golgi cells. Granule cells receive excitatory input from mossy fibers originating from spinocerebellar and pontocerebellar tracts. One of the most important cerebellar tumors is medulloblastoma, a common infratentorial pediatric neoplasm arising from the granular layer of the cerebellar cortex. Their axons ascend to the molecular layer and bifurcate into parallel fibers. Deep to the cortex lie the deep cerebellar nuclei, embedded within white matter. From medial to lateral, these are the fastigial, globose, emboliform, and dentate nuclei, remembered by the mnemonic "Don't eat greasy food." The fastigial nucleus, associated with the vermis, projects to vestibular nuclei and ventral lateral thalamus to influence axial and proximal muscles. The interposed nuclei project via the superior cerebellar peduncle, decussate, and influence the red nucleus and thalamus with final effects on ipsilateral limb muscles due to double decussation. The dentate nucleus projects via dentatothalamic and dentatorubral tracts, influencing premotor and motor cortices and forming feedback loops with the inferior olivary nucleus. All cerebellar output exits primarily through the superior cerebellar peduncle, while afferent input enters through the inferior and middle cerebellar peduncles. The cerebellum is organized into distinct functional zones arranged from the midline to the lateral hemispheres, each linked to specific deep nuclei and motor roles. The vermal zone lies along the midline vermis and is responsible for posture and balance. It receives proprioceptive input from the hips, trunk, neck, and shoulders via spinocerebellar tracts. Output from this zone passes through the fastigial nucleus, which regulates axial and proximal muscles. Lesions of the vermis lead to truncal ataxia, difficulty maintaining posture, and impaired balance. Tumors involving the vermis, such as medulloblastoma, produce difficulty holding the head erect and instability of the trunk. Lateral to the vermis lies the pervermal, intermediate, zone, which controls skilled, voluntary movements of distal limbs. It receives proprioceptive input from the hands and feet via spinocerebellar tracts. Its output is mediated by the interposed nuclei, globose and emboliform. Damage to this zone results in limb ataxia, dysmetria, and impaired fine motor control. Further laterally are the cerebellar hemispheres, forming the lateral zones. These regions are involved in planning, timing, and coordination of complex voluntary movements. They receive cortical input through the corticopontocerebellar tracts and project via the dentate nucleus. Lesions here impair motor planning and initiation rather than basic strength. The flocculonodular lobe forms the vestibulocerebellum. It receives input from the vestibular apparatus via CN8, vestibular nuclei, superior colliculus, and visual cortex. Its output targets the vestibular nuclei, which control eye movements via the medial longitudinal fasciculus and posture via vestibulospinal tracts. Damage causes nystagmus, vertigo, and gait imbalance. Functionally, the thalamus acts as the gateway to the cerebral cortex, relaying and integrating nearly all sensory information except olfaction. Ascending pathways such as the spinothalamic tracts, dorsal column medial lemniscus, and trigeminothalamic pathways synapse in specific thalamic nuclei before projecting to primary cortical areas. Motor signals from the basal ganglia and cerebellum are also processed in the thalamus and relayed to the motor cortex, allowing smooth execution of voluntary movement. Beyond sensory and motor relay, the thalamus plays a key role in arousal, consciousness, pain perception, emotional behavior, and cognition through its widespread cortical and subcortical connections. The thalamus is primarily supplied by branches of the posterior cerebral artery with contributions from the posterior communicating artery. Clinically, thalamic lesions are most often ischemic and produce contralateral sensory loss when the VPL or VPM nuclei are affected. Damage may also cause ataxic choroiditis due to disruption of motor integration. Infarction can result in thalamic pain syndrome, characterized by exaggerated pain responses on the contralateral side. Mass lesions in the thalamus may obstruct the interventricular foramen or cerebral aqueduct, leading to non-communicating hydrocephalus.